Directed evolution of protease enzymes. Directed evolution is a method used in protein engineering that mimics the process of natural selection and to evolve the proteins or RNA with desirable properties not found in nature. The engineering of novel and precise sequence specificity into proteases will provide an important route to the development of existing new tools for analytical, biotechnological, and therapeutic applications. Basically, proteases make up the largest family of enzymes constituting an estimated 2% of all human genes. Some proteases, such as those involved with digestion and protein recycling in cells, are relatively non-selective. Most proteases have evolved to recognize a limited set of peptide sequence, so significant progress has been made in reprogramming protease specificity, for example, using directed evolution as well as other approaches. Proteases have been reprogrammed to cleave substrates containing a variety of amino acids in the P1 and P1- positions. As following cleavage, protease specificity is denoted using the Schechter and Berger nomenclature, in which substrate residues are labeled BX-P1, where P1 is the new C-terminal residue following cleavage, and the residue P1-PX-, where P1 is the new N-terminal. The corresponding subsites within the protease responsible for substrate recognition are labeled as SX, S1, and S1, S1-, SX- respectively. Often, the functional consequence of amino acids that comprise the various protease subsites SX, SX- are difficult or impossible to predict based on the crystal structure information alone. A common methodology for somewhat random diversification of a gene sequence is the error-prone polymerase chain reaction, which is a technique that introduces mutation along the length of the gene being mutagenized. The another technique, which is DNA shuffling, and itchy-scratchy technique. An example of recently engineering proteases using methods such as this are used the Escherichia coli signal peptidase. Space 1. Spaces have been identified and characterized in a wide variety of gram-negative and gram-positive bacteria, mitochondria, and endoplasmic reticulum, and its function is that removes signal peptides from pre-proteins and which has a substrate specificity for small uncharged residues at one position and small or larger aliphatic residues as position 3. In this case, enhanced activity for a desired new substrate was achieved using in vivo. The specificity of SPS1 was reprogrammed to replace alanine and to accept arginine, glutamine, and tyrosine residues at the position 3. Let's see the experimental procedures. SIB1, which is bacterial strain, and the plasmid of Escherichia coli strains, as the following. Respect the vectors as the PET23P vector, which contains the inducible space, and the PONF1 vector, overexpressing pro-OMBA A nuclease A. This nuclease A substrate has a 6 histidine sequence at codons 2 to 7. The second step, which is DNA methods, the quick change mutagenesis method, was used to make oligonucleotides direct mutations within the SPS and the pro-MPA nuclease A proteins. The third, which is purification of a signal peptidase by hashtag. And now let's see the measurement of activity. That's by comparing the practical mass spectroscopy data with theoretical mass. The production of theoretical mass of cleavage products to predict the size of postulated cleavage products to match with the mass spectroscopy data. So you have to use a peptide mass tool on the XPASI proteomics server as shown links. And that's all about our presentation. Thank you, and that's our reference. Thank you.